Bismillahirrahmanirrahim. Assalamualaikum. Good morning, everyone. It's a pleasure, it's an honor, a privilege to be here at the Genomics Summit and to present a very important topic, which is on genomic surveillance of viral diseases in Pakistan. So, as we all know that genomic surveillance, it has played a very key role, especially during the COVID-19 pandemic. And in my talk, I will very briefly discuss some of the progress that we have made at NIH in genomic surveillance of some of the viral diseases, as well as some of the efforts, you can say, or some of the contribution that we have made. So I think the theme of this summit is quite appropriate, that this is indeed a genomic era, and we cannot overstate the importance of genomic surveillance, especially in the field of pathogens and viral diseases. So what does genomic surveillance mean in terms of pathogens? So, looking at the genetic makeup of pathogens with pandemic and epidemic potential, we can look at the evolution of these pathogens, and what it actually does is that it has a direct impact on the development of vaccines, on the development of therapeutics, diagnostics, and similarly on the selection of different public health measures. And I think one of the prime examples of all these is the COVID-19 and the recent pandemic, where we have seen how genomic surveillance basically shaped the pandemic. So these are just some of the NGS facilities that are working in Pakistan. These are not all the facilities, but some of the major facilities, and some of them were very actively involved in doing genomic surveillance of SARS-CoV-2, and the ones highlighted in red are the labs or the institutes that are part of our National Public Health Laboratory Network, and which is being led by National Institute of Health. So all of these 4 labs, they do have NGS facilities. Mostly they have the Illumina systems, and just recently we have established, NIH has established an NGS facility in Balochistan, which is also our provincial public health lab. And so we provided them with Illumina ISIC and with some consumables, so hopefully in a few weeks' time that will be operational. Okay, so these are just the numbers. If you see the numbers of whole genomes of different viruses causing disease in humans, that was submitted to NCDI. So if you look before the COVID-19 pandemic, there are just a handful of viruses, whole genomes that were submitted on this data bank, and most of these sequences, they were done out of the country. But if you see, there has been a significant, you can say, rise in the number of sequences submitted since the pandemic, and the main reason for that is the establishment of different NGS facilities, as I mentioned previously. Same scenario, the number of sequences that are submitted on GISID. So these two, GISID and CDI, are public health databases where pathogen genomes, they are submitted. So again, you can see that in the COVID-19 pandemic, there are a high number of sequences that are submitted. So if we talk particularly about SARS-CoV-2 and the number of genomes that have been submitted to GISID, so you can see that around 7,000 whole genomes have been submitted so far since the start of the pandemic. Our department, it has sequenced around 4,200 whole genomes, and all of them, they are available on this website. Similarly followed by Aga Khan and some of other institutes. So you can just, the purpose to show this slide is to highlight how genomic sequencing facilities, they have been established and what progress has been made since the pandemic started. So yeah, I really wanted to put this slide here. This is basically how the genomic surveillance journey, you can say, started in National Institute of Health. It was very fortunate that I was at CDC in March 2020. It was a training on NGS, and it was for viral hemorrhagic pathogens, especially CCHF. So with the support of CDC and Integrated Global, we received Illumina ISIC. And when I came back, so this is the first run that we processed of NGS in our lab. So this was on 5th of June 2020. And again, I think I must acknowledge the support provided by CDC and the Integrated Global here. So currently now we do have NGS platforms. We have all the three, you can say, major companies. So we have, for Illumina, we have 2 iSeq in the virology department as well as 2 MySeqs. Just recently we have procured the MiniSeq and the NextSeq, which is the high throughput equipment, which is basically the NextSeq 2000. We also have a Thermo Fisher GeneStory S5, and then we also have the 3rd generation Oxford Nanopore technology available as well. There is a dedicated bioinformatic infrastructure and staff that we have, and that is equipped with hardware and software. So we have commercially available solutions to do downstream analysis of these NGS data, and we use in-house pipelines as well. So just recently, we have established a pathogen genomic surveillance center at NIH, and this is going to be, inshallah, a centralized facility that will be used for sequencing not only viruses but for bacteria, for fungi, and for some of the other pathogens as well. And the 2 new Illumina platforms that I mentioned, MiniSeq and the NextSeq, they are basically installed in this facility, and these are some of our team members here. So here I will just very briefly present some of the outcomes, you can say, of our genomic surveillance work that we have been doing since the start of the COVID-19 pandemic. Maybe some of you know that the first variant of SARS-CoV-2, it was called a G614 variant. So there was a mutation in the spike gene at the position 614, and this was basically a mutation which was not present in the original wild Wuhan strain. And this mutation basically led to the virus or the variant to become a more transmissible form as compared to the wild strain. So in March 2020, only 10% of the cases that were reported globally, they had this G614 variant. And in just a 1 month's time, this variant basically spread globally, and its prevalence increased to 70%. And that is just because of this particular unique mutation that you can see in the spike gene. So using our NGS, so we identified this G614 variant in samples that were collected during June 2020. And this was a time then when our first wave was at its peak. Although at that time we had limited genomic surveillance capacity, but we believe that the first wave in Pakistan, especially the peak, was primarily due to this variant. This is another study which was published, and we, using the NGS, we identified the first case of B117, which is also called the alpha variant. I'm sure you must know that. And this alpha variant is basically the first variant of concern, and it had around 25 mutations compared to its ancestor. And it was identified in September 2020 in UK, and then it rapidly spread to other parts of the world. So we identified this variant in travelers that returned from UK to Pakistan, and we also published this data. So this was again another study using next generation sequencing, where we did surveillance of the B117 or the alpha variant. And I think this is a very classical example of how we have used genomic surveillance for public health action, so from a public health point of view. Because if you look at the graph on the top, you can see that the second wave in Pakistan, it was on the decline in December, January, in basically January and February 2020. And this is basically on the basis of the national case numbers. But if you look at the second graph, and this is our genomic data, it showed that the alpha variant, it was increasing exponentially. So in January, there were very few cases, but in February, you can see the exponential growth of this alpha variant. So based on our lab data, we basically informed our public health experts, especially at NCOC, because I used to present this data at NCOC. So we very timely informed NCOC that the variant is basically spreading. It's emerging in Pakistan population. And what it did, it gave them time. It gave them time to prepare. It gave them time to prepare for a possible third wave. And this is exactly what happened. So just after 1 month, you can say, or maybe around 5 weeks, there was a third wave. So this is how we have been using our genomic sequencing data to inform public health decisions. This is again another study published of our genomic sequencing that we did during the third wave. Although the third wave in Pakistan was due to the alpha variant, but there were some other variants circulating at that time as well, such as the delta, gamma, and as well as the South African variant, the beta variant, and all these variants they were also called as the variant of concern. So here in this study, our focus was not on alpha variant, but we wanted to see what other variants are circulating in the country. So we selected only those samples that were negative for alpha variant. And notably, interestingly, we found a high number of cases of delta variant as well here. So this is another study where we did sequencing, and we found that the fourth wave in Pakistan was caused by the delta variant, also known as the Indian variant at that time. And this is again a highly, you can say a variant which had a lot of mutations compared to the previous variants, and there was some mutation that had implications on disease severity as well. So we also very timely, using the genomic surveillance, we very timely reported not only the emergence but the spread of this variant in different geographies of Pakistan. And again, this data was presented at the national level at NCOC. This is our latest or the recent work that was again published in one of the journals. So here we, using our NGS, we looked at the different variants that were prevalent in the country during the 6th wave and the post-6th wave. So we came to find out that a number of Omicron subvariants or sister lineages, they were infecting people of Pakistan. And the first Omicron variant that was imported into Pakistan was the BA.1, and then it was replaced by BA.2, and similarly XBB. So all these, there are a plethora of variants when we talk about SARS-CoV-2. So we did genomic surveillance of these variants, and we again reported these variants to the concerned authorities. So moving from SARS-CoV-2 to some of the other respiratory pathogens, we have leveraged the NGS facility that was established during the COVID-19 to do genomic surveillance of some of the other viruses as well. And this is one example where we sequenced the whole genome of mumps virus. Back in 2022, there was a huge outbreak of mumps in Pakistan. Maybe some of you may know that. So there were more than 20,000 cases reported. And that outbreak, it continued into 2023, into the first 2 months of 2023. So this was a study that involved samples from 2023. So what we found that the virus that was prevalent was the G genotype. And basically, so mumps, there are different genotypes, and when we compared our strain with the vaccine strain, which is the Genomic strain, it has the A genotype. So we found a lot of changes and divergence when we compared it to the vaccine strain. And again, I think this has unique, very important implications, especially for our vaccine program, because in Pakistan, in our national immunization program, we have the measles and rubella vaccine that is part of our EPI. But if you look at the global countries, MMR is used, so they include mumps as well. But that mumps vaccine, it has the antigen of the Genomic strain, which is a lineage A or a genotype A. So our study showed that the genotype G is circulating. So again, it has implications on vaccine design in future as well. We also carried out whole genome sequencing of RSV, respiratory syncytial virus. Again, this is the first study from Pakistan. We did retrieve whole genome of this virus from the country. RSV, in case you don't know, is a major cause of lower respiratory tract infections, especially in children. and it is a major public health issue for children, for pregnant women, as well as for elderly above 65. So, but from Pakistan, we don't have any data. Before the study, there was no data available on the genetic diversity of the viral strains that were circulating in the country. So we again used NGS, and we managed to sequence the virus. So it has the two surface glycoproteins, the G and the F proteins, and both they are involved in attachment to the host cell. And so based on our mutation analysis, we found several unique mutations. And again, this has implications on viral fitness when we talk about from public health point of view. So coming from some of the respiratory viruses, I think this is again a very latest example of how we have been using genomic surveillance from public health point of view, how we can take actions on this data. So just recently we had an outbreak of dengue, especially in Rawalpindi and Islamabad, causing severe cases in the primary infection. And normally most of the severity associated with dengue is in the secondary infections. So there was a concern raised by health authorities whether the virus has changed or there are some mutations in the virus that is causing severity on primary infections. So we provided the answer to this question based on our NGS data, and we found that the dengue serotype 2, it belonged to the cosmopolitan genotype, and the clade was the 4A clade, and there was actually no such mutation or change when we compared this strain to the previous strain that were reported in 2021, 22, and 23. And similarly for dengue 1, DEN1, we also found that the strain was quite similar to our previously circulating strains. So I think, again, providing timely information to the health authorities, I think that is the main purpose of genomic surveillance, and that should be in fact the purpose of genomic surveillance for viral diseases. Another example here, we have done whole genome sequencing of chikungunya virus, and these cases or these samples from these cases, they were referred from KP region. So based on our whole genome sequencing data, we found that the ECSA, the East Central South African genotype, was prevalent. Interestingly, there is a particular, so chikungunya, it's spread through Aedes mosquitoes. So Aedes aegypti and Aedes albopictus, but primarily it's spread through Aedes aegypti. And there is a unique mutation in the envelope 1 protein, which is at position 226. If that mutation takes place, then the virus can adopt from Aedes aegypti to Aedes albopictus. So that can have severe implication and complications for any public health system, especially for countries like Pakistan, where both vectors are prevalent. Fortunately, in our sequence strains, we did not find that mutation. So another vector-borne virus, Crimean Congo hemorrhagic fever virus, another disease, a highly lethal disease. It's common in Pakistan, especially our Balochistan region. It is a hotspot for this disease. So we did a whole genome sequencing study, and this was from 2017 to 2020. So we generated 36 whole genomes of CCHF from Pakistan. Again, I don't remember previously whole genomes of this virus available from Pakistan. And we again came to find some unique changes. So the main lineage was the Asia 1 lineage. And we found, because this is a segmented virus, there are 3 segments, small, large, and medium. So there are possibilities of reassortment. So we found some reassortments in few cases as well. And again, I think this study was done using the IC platform that we initially got from the CDC. We have also done similar study for our 2022 CCHF, and again this data is available on different platforms and published as well. So moving to Mpox, I think we all know Mpox has been declared as a public health emergency of international concern. In fact, it has been designated this twice in a limited period of time. So we started doing genomic surveillance of Mpox back when it was in 2022, when it was first time declared as a public health emergency of international concern. But this data is from 2023, where we sequenced the first whole genome of Mpox from Pakistan, and we did this in 48 hours. My team is here. So we did this in 48 hours, and we came to find that this was basically a clade 2B variant. But fortunately, this was one of the variants that was less lethal as compared to clade A. So clade A has a mortality of up to 10%, whereas clade 2 has a mortality less than 1%. So the 2022-23 outbreak of Mpox in fact in globally was caused by the clade 2B. That's why we saw very few deaths during the 2022-23 outbreak. However, in the current outbreak, which is at the moment mainly confined to the Democratic Republic of Congo and some of the neighboring countries, it is caused by the clade 2B, clade 1B, and which is obviously more virulent, which has higher fatality rate. But so far, we have sequenced around 6 whole genomes of the Mpox virus, and we have not detected any clade 1B variant in Pakistan. And again, all these cases, they were imported. They were imported either from the Gulf countries or some of the Arabian countries. And interestingly, using our genomic sequencing, we found that the Mpox clade that we found was the A.2.1 lineage, which is a very, very rare lineage, and very few sequences of this lineage have been deposited on NCBI. So to us, getting this lineage, and the last time this clade was detected was in India, and it was back in 2022 in May, June. And again, we have been using this genomic surveillance data to inform our public health authorities, especially the NCUC. Okay, so apart from doing the genomic surveillance and looking at how the virus or pathogens they are evolving in Pakistan, what variants are circulating, and how and where they are spreading, we have also used our NGS to identify different pathogens. And we have done that using our metagenome approach. So back in 2023, in September, I think there were a huge number of cases of acute hemorrhagic conjunctivitis, also known as pink eye, reported in Punjab. And if I'm not wrong, there were around 400,000 cases that were reported. But the etiological agent of that outbreak was unknown. And so we used this, so we used metagenome approach, and we identified that it was caused by a Coxsackie 824 variant. So Coxsackie viruses, they have been associated previously as well with acute hemorrhagic conjunctivitis cases. And so again, you can see the role and the importance of next generation sequencing, importance of genomic surveillance. Here we used it quite effectively to identify the causative agent of this outbreak. And again, back in 2023, we received a sample from an encephalitis patient from KP, one of our tertiary care hospital from KP. And again, the agent or the pathogen, the positive agent was unknown. So again, we applied our metagenomic approach, and we managed to retrieve the whole genome of West Nile fever virus. Again, this is the first time sequence of this virus has been retrieved from Pakistan. Previous studies are available, but they are mainly focused on the serological data or PCR. And if you know, just for the information, so this West Nile fever, it is spread through Culex species. So we do have this mosquito or the vector in Pakistan. So having a facility that can quickly identify such viruses, and knowing the fact that this vector is already prevalent in the country, I think it plays a very key role when we talk about in terms of our public health system, and especially the surveillance point of view. Okay, so we have been sharing our genomic data not only through publications, but we have been sharing our genomic data through a number of different resources. So we have made preprints. So preprints, I guess you know that preprints are not peer reviewed, so any useful finding we always used to write a preprint, and we used to make it available for the scientific community. We have been uploading sequence data to the global data banks. And importantly, we have been making or preparing these situation reports on genomic surveillance of different diseases such as COVID, dengue, and mpox. And all of these situation reports, they are available at NIH website. So if anyone is interested, they can just log on to the site and they can see it. So we also, our bioinformatics team, they are also maintaining the next frame build. So we have data available on this platform as well. I think I would like to highlight here, back in 2023, in February, March, NIH, along with the National Genomic Consortium for SARS-CoV-2, we developed a national genomic surveillance strategy for priority disease of Pakistan. How many of you know that there is such a strategy? If you can raise your hand, I would like to know. So very few people, okay. So we developed this strategy back in March 2023, and obviously relevant stakeholders were part of this strategy. And Pakistan, by the way, is one of the few countries that have developed this strategy. And obviously, without a strategy, it's very difficult to do genomic surveillance for priority diseases. And let me tell you, we are in the process of updating this strategy. We have done some site visits across the country to look at the gaps that are present in our sequencing facilities and what are some of the limitations and what are some of the strengths. And we have also, this time, we will be including our animal sector as well. So this will be a One Health genomic surveillance strategy for priority disease of Pakistan. So hopefully we will be completing this task very soon. Again, this strategy is available on NIH website. If you are interested, please do have a look. So just lastly, there are a number of challenges that I think previous speaker has also mentioned. So we do have challenges of sustainability. And yes, it's an expensive technology. So sustainability is a problem, especially after the COVID-19 pandemic. A lot of the funds have been withdrawn. So that has an impact on reagents, on retention of HR. And another challenge is the data integration. I think just the genomic data provides a very limited information. So it's important how we integrate our clinical data, our epi data with our genomic data. And if we can do that, I think that is the time where we will be doing in real-time surveillance of these, genomic surveillance of these different pathogens. And as my colleague mentioned, so there is again a limited coordination between our public health laboratory network as well as our academia, and how we can address this. So obviously, I think there has to be ownership from our stakeholders, from the government. These are national programs, so this has to ownership has to be taken by our health authorities. We need to strengthen our national laboratory network, and there should be dedicated funds for this genomic surveillance activity. And lastly, again, I think this is again a great platform to interact and to communicate and make linkages. So we need to build linkages with academia as well. And lastly, so I would like to acknowledge all those partners that have supported us during the pandemic for establishing and then strengthening our genomic surveillance facilities. So I will basically conclude with just a couple of lines. As mentioned, I think previously, so genomics, it's not just now a tool. I think it has become a necessity, it's a reality for public health systems. Without this, it will be very difficult to control different diseases in future as well. So I think we as scientists, as people who are working in this area, we can always leverage and we can always cooperate and collaborate with each other and use our experience, our expertise, the capacities that we have to strengthen genomic surveillance, not only for viral diseases, but for cancer and for other aspects as well. Thank you so much.